Okay, so I'm going to go through some of these PowerPoints pretty quickly because I did go through this in class after the exam. Um, let's see. So we talked about the orientation of the kidneys, where they're located. Uh, we looked at where the other major organs, the ureters, the bladder, the urethra, where they're located, and you should know their functions. I'll go ahead and quickly kind of point out some of these structures again. So when we're looking at the kidney, this outer tough fibrous capsule, that's called the renal capsule, and that helps hold, give the kidney structure, as well as it plays a protective function because again it's retroperitoneal. So it's actually behind the parietal peritoneum, right up against the back muscles, so it needs as much protection as it can, and that's why you find all that adipose and renal fascia surrounding the kidneys as well. So we have the renal capsule. Now when you look at the kidney, you can see that there's this outer layer. This is called the renal cortex, and then this inner, which is the renal medulla. When you look at the medulla, you can see that there's these areas, this slightly darker area. They look like little pyramids, and those are the renal pyramids. So these are the renal pyramids, and this lighter tissue that kind of dips down in between the renal pyramids, those are called the renal columns. Where the renal pyramids come to an apex, this is called the renal papilla. Okay, so this little apex of each pyramid is called a renal papilla. These structures that the renal papilla empties into, these are the minor calyces, and then the minor calyces merge to form a major calyx, and then multiple major calyces merge to form what's called the renal pelvis, and that renal pelvis exits and narrows as the ureter, and then that ureter is what's going to transport urine to the bladder for storage. Now, where it doesn't show the renal artery and renal vein, but this would be, the renal artery would be entering here, the renal vein exiting here. So where this everything is entering and exiting the kidney, this region is called the Hylas, or the hilum, either or. Now, if you look at where the minor calyces and the major calyces are, there's this space, right? And that space is usually filled with adipose to help hold its structure and give cushion and protection. And that space, that is where everything is going in and or surrounding the minor calyces and the major calyces. This space is called the renal sinus. Okay? So, and when we talk about the formation of urine, all of the filtration of the blood, that is going to happen. The filtration, reabsorption, secretion, that's all going to take place in the structures that you find in the renal cortex and the renal medulla. And then once what is destined to become waste, once it exits the renal papilla and enters into the minor calyx, it will be eliminated as urine. And so the flow would be from cortex through the medulla, through the renal papilla, through the minor calyces, the major calyces, the renal pelvis, and the ureter out. Remember I said that you can classify, or you can group all nephrons, which nephrons are the functional units of the kidneys, up to approximately 1 million nephrons per kidney. And all nephrons can be grouped as either being cortical or juxtamedullary. And if we look at this image right here, this is showing you this lighter pink would be the cortex, this darker pink would be the renal pyramid. Now, this nephron, which is primarily located in the cortex, this would be a cortical nephron. This nephron, which is located in the cortex, but this loop dips really far down into the medulla, this is a juxtamedullary nephron. Now, both have the overall same functions, but it's these juxtamedullary nephrons which are going to be extremely important in maintenance of water balance. And so when we look at the nephron, we can group it into the regions called the renal corpuscle and then the renal tubule. And the renal corpuscle is made of a Bowman's capsule, which has two layers, a visceral and a parietal, and the glomerulus. And the glomerulus is a specialized tuft of capillaries, which is going to carry out the glomerular filtration, and filtration is the first step in urine formation. And then the renal tubule is going to carry out the reabsorption and the secretion. So here, this ball, right, and when you look, this little ball-like structure here and here, those are renal corpuscles, okay? And so that outer parietal layer is this simple squamous epithelium. This is, we also refer to it as the Bowman's capsule, so this is the parietal layer, the Bowman's capsule, simple squamous epithelium. And then if we look inside this little tuft of capillaries, this is the glomerulus, but there's these cells that are directly adhered to and surrounding the capillaries, and these cells are the podocytes, and those podocytes make up the visceral layer of the capsule. So that would be the visceral layer, and this outer simple squamous epithelium is the parietal layer. So this vessel coming in, this is what's called the afferent arteriole or afferent arteriole. This is what is bringing blood into the capillary bed, right? And this vessel leaving, you can see the arrow coming out. This is the efferent or efferent arteriole, and this is where blood will be leaving the capillary bed. So it comes in through the afferent arteriole, circulates through the capillary bed, and leaves through the efferent arteriole. Now, I said coming in through an arteriole, capillary bed, leaving through an arteriole. Normally, when we talked about the systemic capillary beds, it was arterial, capillary, venule. Well, the fact that we have an arteriole coming in and an arteriole leaving a capillary bed, this helps maintain a relatively high pressure within this capillary bed, because this is where filtration takes place, and that's the first step in urine formation. And filtration requires a pressure gradient. So we have to maintain an increased blood pressure in this capillary bed to drive filtration. So when blood comes in and there's a high pressure within this capillary bed, it pushes water and other small solutes into this space, which is called Bowman's space. And this outer parietal layer of the Bowman's capsule, this is what collects what is now called filtrate. So when water and other small molecules and solutes, when they get pushed out and collect in this space, it's now called filtrate. It's no longer part of the blood plasma, it's a part of this filtrate, which fills this area, okay? So arteriole coming in, arteriole leaving, keeps blood pressure high. It means that the glomerular filtration rate will maintain, will stay where it needs to be. And if you notice that the afferent arteriole has a larger diameter coming in than the efferent has leaving, this larger diameter allows more blood to come in, then the smaller diameter means less blood can leave, right, at the same time. And so again, another way to maintain a high pressure within this capillary bed. And the glomerular capillaries are unique because they're fenestrated capillaries. Fenestrae means that they have holes in them, so they're more porous than other systemic capillaries. And so these fenestrae allow larger molecules to get in, to get out. Because the point of these kidneys is to filter blood, let things get out into that filtrate. So things that, but it's still, it's not going to allow whole cells. It's not going to allow large proteins. Maybe a little bit of albumin can get out because albumin's a relatively small plasma protein. But for the most part, it's going to be water and small ions and things like glucose that can exit and become part of the filtrate. Now these podocytes, which make up the visceral layer, these are important in infiltration, because if you see where you see the cell body and the nucleus, they have these long cellular extensions that come off of the cell body, and then multiple little feet-like processes that extend off of that, and those are called pedicels. And then what happens is the pedicels from multiple podocytes will interdigitate and kind of form like a woven type pattern, and this creates slit pores. So this is helping create a size exclusion. Only those molecules that can fit through the fenestrae and then fit through those slit pores will end up as a part of the filtrate. So this makes sure that whole cells and really important proteins aren't going to be able to make their way out. They'll stay a part of the blood plasma. Okay. Now, so blood has come in, we maintain a high pressure gradient in this capillary bed. This forces water and other small molecules out into the filtrate, and then that filtrate, as it collects into this Bowman space, will eventually exit through this first part of the renal tubule called the proximal convoluted tubule. So this is the first part of the renal tubule called the proximal convoluted tubule, and proximal because it's closest to the renal corpuscle. And you can see it's simple cuboidal epithelium, and it has microvilli, and that's important. So here this would be our renal corpuscle with our arterioles coming in and leaving, our glomerulus. This green would be our proximal. See how it's highly convoluted? So proximal convoluted tubule. And then once it straightens and dips down, whether it be a little bit into the medulla or a great distance into the medulla, this is the loop of Henle. And so this would be the descending loop of Henle. This would be the ascending loop of Henle, and you can see that there are thinner regions, which would be like a simple squamous, and there are thicker regions, which would be more of a simple cuboidal. And then once it gets back into the cortex, so if I drew a dotted line across here, right, the proximal and the distal convoluted tubules would be primarily located in the cortex, and then the loop of Henle would be what dips down into the medulla. So descending loop of Henle, ascending loop of Henle, and then this purple is the distal convoluted tubule, distal because it's not directly attached to the renal corpuscle. And then this distal convoluted tubule connects to this structure, which is called a collecting duct. And a collecting duct, an individual collecting duct, isn't a part of any one nephron, because you can see these little extensions. This would be multiple distal convoluted tubules coming and connecting to a single collecting duct. Now, where are those collecting ducts located? Let's go back to this picture. You see the renal pyramids, and they're very striped. They have a striped appearance. That striped appearance comes from the fact that all these collecting ducts from all of these nephrons come down through the medulla and will ultimately collect at this renal papilla, and everything that's flowing through those collecting ducts, give or take maybe some water that gets reabsorbed, but as the filtrate flows through these collecting ducts, it collects at the renal papilla, exits the renal papilla into the minor calyx, into the major calyx, into the renal pelvis, and then out through the ureter. So that striped appearance of the renal pyramids is due to all of those collecting ducts traveling down and meeting at the renal papilla.